All right, so looking at these two chapters here, we got two sides of the same coin. Okay, so every one of us basically has two immune systems, if you will. You have an innate immune system and then an adaptive immune system. Okay, your innate immune system is your passive, non-specific immunity. Okay, we all have the same version, and it's always protecting us, keeping foreign entities at bay. So, for instance, we all have skin, we all have mucous membranes, we all have a microbiome, we all have white blood cells, we all experience fever and inflammation. Okay, these are all parts of your innate immune system. Okay, non-specific, passive, always there, always protecting you. We all have the same version. Now, these things can be compromised. Okay, so that's when your adaptive immune system can swoop in. And calling it adaptive is the best term for it because your adaptive immune system is the part of your immune system that customizes itself to infection. It learns from infection so that if you encounter the same pathogen again, it knows what to do more quickly. So your adaptive immune system is why vaccines work. Okay, you're educating your immune system, teaching it how to deal with the pathogen without ever having to experience it that first time around. So it does retain a memory. Now, unlike your innate immune system, though, every one of us have slightly different adaptive immune systems. Okay? It customizes and adapts itself to all the crap you encounter throughout your lives. And we've all encountered different crap, and so you'll have slightly different libraries, if you will. So another way of looking at this as 3 lines of defense. So your innate immune system makes up first and second lines of defense. Okay, first line of defense would be, oh, no. That's not good. I'll make it for fun editing later. Okay, so your innate immune system starts with the first line of defense. Okay, physical, chemical, microbial barriers. Okay? If those things are compromised, say you injure yourself, you cut yourself, then comes your second line of defense. Okay, white blood cells, cellular defense. Okay, they go to work, they phagocytize foreign entities, but even still, they can only do so much. Okay, so if the pathogen is outpacing your white blood cells, then you have to rely on your third line of defense, your adaptive immune system. Okay, B cells and T cells. Okay, antibodies are a part of your adaptive immune system. So, Chapter 16 is all the good innate immunity stuff. Chapter 17 is going to be adaptive immunity. All right, so we're going to look at first lines of defense to start, and then we'll get into the rest of it being mostly second lines of defense. So we'll talk about how your immune system communicates. We're going to talk about sensor systems. So how does your body identify that there's something inside of you that doesn't belong there? We'll touch on the phagocytic white blood cells and how phagocytosis works. And then we'll get very briefly into inflammation and fever, concepts I rely more on the phys teachers to hit harder on. Okay, so looking at your first line of defense, okay, keep it simple. You've got your physical barriers, we've got our own special brand of antimicrobial substances, and even our microbiome, our normal microbiota, is a part of our immune system. Okay, so looking at those physical barriers, I love this diagram. It's from another text. It's an oversimplification of the human body, showing that we are nothing but watertight, sealed up meat bags. Okay, there are no gaps. So in beige, you have skin. In pink, you have mucous membranes. Okay, at all the ports of entry, there are no gaps in these tissues. Okay, they are connected to each other. So we are surrounded by a mostly impenetrable barrier. And I say mostly. So your skin is truly antimicrobial armor. Okay, it is not a happy place for most bacteria. So, why is it antimicrobial armor? Well, it is immensely difficult to penetrate on the microbial level. Okay, it is fairly thick. You've got 2 layers, right? You've got your dermis and your epidermis. So at the base, you have your dermis, made up of tightly woven connective tissues. Fairly thick, fairly dense, hard to penetrate. Above that, though, you get to that epidermis, where you have all those many, many, many layers of thinning epithelial cells. Okay, and they're constantly pushing up and out. So as you get to the top layer, the cells are fairly old, dead, thin, and they flake away as you brush it. So this is, alone, very difficult to penetrate for a microbe. Okay? Those that can penetrate this either do so because you've injured yourself, or they're specialists. They get in through hair follicles, pores, that sort of thing. Okay, so looking at the surface of your skin. As a microbe, skin is a very dry and salty, inhospitable environment. Okay, so most of the bacteria that are on your skin, the microbiome you have here, they're gram-positives. Okay, harmless, like Staphylococcus epidermidis. They're tolerant of dry conditions and salty conditions. Okay, you have very little gram-negatives on your skin. So your skin is also watertight. Okay, it's embedded with keratin. So again, it's difficult for microbes to establish themselves. Okay, on the surface of your skin, honestly think about it. You do have a lot of gram-positives, but do you think they're going to be on the drier parts like your forearms, or more like armpits? Right. They're going to be in the areas that tend to be sweatier, in the stinkier areas, thanks to those organisms. All right, so skin is, like I said, mostly impenetrable to bacteria. Now, your mucous membranes, on the other hand, are just a wee bit more sensitive. Okay, so we're talking about your genital tracts, GI tracts, sinuses, your lungs. Okay, basically, these are areas where we're all familiar with infection. Right? Sinus infections, chest colds. There's tons of STDs out there, right? UTIs. Okay. These are the most commonly affected areas in your body because mucous membranes don't have a ton of protection. And part of that is because of the structure of your mucous membranes. That histology, you know all too well, that your mucous membranes are one cell layer thick. Okay? Just one sad layer of columnar epithelial cells. They're very tall, but they're not particularly thick. So, they are fairly easy to penetrate for certain bacterial pathogens. Now, mucous membranes aren't without their own needs of protection. They're called mucous membranes. Okay, mucus, while unpleasant, is effectively a sticky glue trap for bacteria and basically anything else you breathe in. So I love this image. And I couldn't believe this was an actual SEM when I saw it, because it's colored after the fact. You have a cross-section of a goblet cell. Goblet cells produce the bulk of your mucus. And on the surface here, it's probably a lung sample. You got a lot of ciliated cells covered in this greenish mucus. I love they chose green. And you get this probably pollen particle trapped. So anything you really kind of breathe in gets caught up in this mucus. And then, once it's trapped in the mucus, well, three things can happen. Right? It's spit out, it's swallowed, or it's shed as feces. Well, I guess you can throw sneezed in there. But sneezed and spit, they kind of fall under the same umbrella. Alright, so mucus is a sticky glue trap. So like I said, as you breathe in pathogens, a fair amount of mucus does get swallowed. So what goes in must come out. So anything you swallow is pushed through you through peristalsis. Right? You have involuntary muscles that undulate your esophagus, stomach, small and large intestines, and colon. So for many bacteria, when they're swallowed, they don't survive passage through the stomach. They get destroyed by your stomach acids. Okay? This is actually a challenge for a lot of probiotics because a fair amount of microbes you ingest actually do get destroyed before they get to the destination. Now, your lungs are a different story. Okay? Your lungs are a part of your body that is fairly sterile, devoid of microbial life, because of how sensitive it is and how close to your bloodstream it is. So your lungs are constantly moving out all of the crap you're breathing in every second. Because every time you breathe, it is filthy dirty air. As it enters your lungs, the ciliated cells in your lungs, and cilia being these little hair-like appendages, are constantly pushing all that material back out. So they get trapped up in mucus. And this is what we call the mucociliary escalator, how your lungs keep themselves clean. So, while your mucous membranes tend to be fairly sensitive to infection, they aren't without their own defenses. Now, the next thing we're going to look at is a short list, because there are other examples, of your own brands of antimicrobial substances that you make. Okay, so, we don't make antibiotics, but we do make other effective means of combating bacteria. So, for instance, on your skin, okay, besides being dry and arid, it's also somewhat oily, right? On our skin, we produce sebum, okay, a fatty acid that is fairly toxic to a lot of bacteria, especially gram-negatives. So it will embed itself within the gram-negative, causing them to become leaky. Now, lysozyme. This is an enzyme that just keeps coming back. I think it's the third or fourth time we've mentioned lysozyme. Lysozyme is the enzyme that breaks down peptidoglycan. And we as humans produce lysozyme in all of our excretions. Saliva, tears, mucus. So to make this relatable, let's talk about eye infections. Sorry. The skin around your eye is covered in gram-positive bacteria. Yet, our eyes are not being infected all the time. Okay? So, that is in part thanks to lysozyme. Okay? The lysozyme is constantly killing off gram-positives that get too close to your eyes. Right? They are just naked peptidoglycan, easily destroyed by lysozyme. But say you get conjunctivitis, some manner of pink eye. Odds are, if it's not viral, it's probably a gram-negative pathogen. Okay, they've got that outer membrane, right? The outer membrane makes them resistant to lysozyme. So, while you are covered in gram-positives, okay, they don't infect your eyes constantly because of, in part, lysozyme. Sorry. All right, so iron-binding proteins. This is something we'll talk about a bit more in the last week of school, but many pathogens need a fair amount of iron. Okay? And all cells need iron. It's a cofactor. We need it for our enzymes to work right. Many pathogens just need a lot of it. So when you have a pathogenic infection, in many cases, the pathogen has to go searching for iron. And where are they going to look? Well, probably your bloodstream. A lot of iron. Your body can react to this. It can say, all right, I'm just going to hide any free iron. So you make proteins like transferrin, lactoferrin, ferritin, proteins that will actually snatch up the free iron and attempt, use the word attempt here, to hide that iron. Now, why I say attempt is because, and we'll talk about this more in the last week of class, there are some pathogens that produce a protein that just steal it right back. But your body does make its efforts to hide the iron and starve out the pathogen. And then, and I should look up some good examples, so I'm not as familiar with these. We make antimicrobial peptides. Okay, so think a really short protein with a short string of amino acids, upwards of 50 amino acids long. So it's not a functional protein. Proteins are hundreds of amino acids. This is a little string of amino acids that basically attack bacterial cells. They embed them in the they embed themselves in the cell, causing it to burst, causing it to lyse. All right, so that's our own brand of antimicrobial substances. Okay, and it's a short list. We do make other things as well. Now, the last part here is something that has been a newer revelation. Okay, as we have tried to understand the role of our microbiomes, the friendly bacteria we have in our bodies, one of the first things we did establish is that your symbiotic microbes, your microbiome, is a fundamental part of your immune system. Okay? We all have a microbiome, and it is all there in part to help protect us from pathogens. So, how do they protect us? Okay? Well, ignore your skin. Your skin doesn't have a huge microbiome. What does, though, is your GI tract. Okay? Most of your bacteria are found within your GI tract. Areas that tend to be a bit more sensitive to infection. So, one of the things they do for you is competitive exclusion. You have so much bacteria lining your intestines and colon that there's no free real estate. Pathogens need space to establish themselves and colonize. There's not a lot of extra space to be had. And on top of that, they gobble up the nutrients that bacteria would normally want. So there's not as much food available either. Now this is where it gets kind of wild, and they're coming up with new examples all the time. Your microbiome, your friendly bacteria, also make their own special brand of antimicrobial substances. So going above and beyond what we make, they make things that are kind of like antibiotics, but not the same. Okay, so we're not talking about antibiotics. So some examples. Propionibacterium. You're all familiar with propionibacterium, whether you like it or not. If you've ever eaten cheese, you've eaten propionibacterium. Now, this doesn't exactly relate to this, but maybe you've had Swiss cheese. Okay, it's got that unique tang to it. Or maybe you've had cheese where it's starting to kind of crystallize inside. Odds are that's propionic acid from the propionibacterium. So these are an important part of your microbiome. We all have them. And one of the roles they play is actually produce toxic fatty acids. Harmless to us, but harmful to a great deal of gram-negative bacteria, or gram-negative pathogens. E. coli makes something especially odd. E. coli produces what's called a colicin. Okay, so break down the term here. Coli, as in E. coli, and then sin. Right? You attach CIN to any word, you're killing something. But it's a coli killer. Not exactly. Colicins are compounds that will attack and kill close genetic relatives of E. coli. It kills its cousins. So we talked about all those enterics, the coliforms, like Shigella, Salmonella, Klebsiella, Proteus species. Colicins would kill them. Okay, so if you ever have a Salmonella infection, your E. coli population is helping you out and combating. And then the last line applies to women. The vagina plays host to a great deal of lactobacillus that like a good acidic environment. Okay? So the vagina actually has a severe advantage of being acidic. Pathogens are neutrophiles. They don't establish well in acidic environments. And there's others. This I'm going to replace. This is the last semester. I swear I'm going to show this diagram. There's newer, better ones out on the internet. This is just showing you that the various parts of your body have different microbial populations. I have a supplemental lecture I'll try to fit in the last week where I actually get into some detail about the microbiome. Okay, so those are your physical barriers, right? Skin, mucous membranes, antimicrobial substances, microbiome, all of which can be compromised. So I always leave an example of you accidentally cut yourself. So unless you cut yourself with something totally sterile, odds are you're going to make that wound dirty, meaning bacteria will get in there. So what comes next to defend you? Your cellular defenses. In short, we're going to talk a whole lot about white blood cells. All right, so the bulk of your blood cells are restricted to two places, your blood and lymph, circulatory systems and lymphatic systems. And blood cells, if you're unfamiliar, all derive from the same cell, hematopoietic, a type of pluripotent stem cell. And there's a handful of different stem cells in your body. Okay, these pluripotent stem cells, they are found in your bone marrow. And it is their job to mature and become all of your various blood cell types. Okay, so, disclaimer, do not memorize this. This is more appropriate for like a phys class or even an immunology class. But, just want to show you folks how this happens. So, somebody won a lot of accolades for sorting this out. But this is the cascade of events that occurs between stem cell and all of your blood cells. So like I said, in your bone marrow, you've got these pluripotent hematopoietic stem cells. They receive chemical signals, which we'll talk about later on, telling them which route of maturity they should take. So, often they become myeloid stem cells. And myeloid stem cells become the bulk of your blood cells. So, red blood cells, platelets, and various other white blood cells. Now, on the other hand, if they become lymphoid stem cells, well, then they turn into the cells involved in your adaptive immune system. B cells, T cells, natural killer cells. But we'll talk a little bit more about how and why this happens in just a little bit. Okay. So looking a bit more into some of these white blood cells. Okay, I need to get red blood cells out of the way. We won't talk about them much. They carry oxygen throughout your body. We call them erythrocytes. Great. All right, so white blood cells, leukocytes. You have two major groups of white blood cells to be found: granulocytes and agranulocytes. The difference between them overall is whether or not they have biologically active granules inside of them. These granules show up when you use a particular stain under a microscope. But aside from that, each type is going to have its own specific role. All right, so let's look at the three types of granulocytes. First, we've got neutrophils. Neutrophils are really, really important initially during infection. Because while if you're not sick, you're not making very many of these, if you are sick, you're making a lot. Okay, so your body will produce mostly neutrophils during an infection. Okay, they are the primary soldiers during infection because of what they do. They are phagocytic. Now, a little hand waving here because I'm going to get to phagocytosis in just a little bit. Okay, but as a spoiler, phagocytosis, if you're unfamiliar, is the engulfment and digestion of foreign material by your cells. So neutrophils find bacteria, engulf them, and then digest them. They kill them. I bring up the fact it's phagocytic because some of your white blood cells are phagocytic, some are not. So neutrophils are very phagocytic and produced primarily only during infection. Basophils. Basophils are not phagocytic. Their role is more specialized. They are primarily involved in inflammation. Okay? They're really good at making histamine. So basophils, when they're not phagocytic, they're out there searching for things that don't belong. So if they detect that that wound is injured, they're going to cause inflammation. All right, so eosinophils. Eosinophils are phagocytic, but they're highly specialized. The only time you'd be making eosinophils in mass is if you had a parasitic infection. We're talking protozoans or helminthic worms. Okay, they're really good at dealing with parasites. So those are our granulocytes. We've got the neutrophils, the basophils, and the eosinophils. Now, the agranulocytes here, we got a few more. Okay? Some of them are a little bit more specialized. So the first example is mononuclear phagocytes. So we've got monocytes and macrophages. So why do we lump them together? Well, take a look at this cascade. Monocytes, macrophages. Macrophages are derived from monocytes. So why are we talking about both? Well, it's because where they're found in your body. Monocytes are restricted to your blood and lymph. Okay? They do not leave the circulatory system. They do not leave the lymphatic system. Macrophages, on the other hand, have free reign. They go wherever the hell they want. Okay? So they're found primarily, say, outside of your blood and lymph. They can actually scavenge kind of everywhere throughout your body, which will be important in a bit. Both of these guys, though, are phagocytic. Dendritic cells. Dendritic cells we'll see again in the next chapter, because they bridge the innate and adaptive immune systems. So a dendritic cell will go to that injury you have. They'll go into that cut, and they're phagocytic. They will sample some of the bacteria in the wound. digest it, and they're going to take all the little bits and pieces of the bacteria, like gruesome trophies, and they're going to take all those little antigens from the pathogen and take it all the way back to your lymphatic system. Okay, this takes a couple of days. Once they get to your lymphatic system, they look for your B cells and T cells, because they want to show your adaptive immune cells, hey, did I just find something bad? Should we do something about this? Okay, that'll make more sense when we get to chapter 17, but they bridge the two immune systems. They go and sample pathogens from a wound, and they bring them back to your adaptive immune system for verification. And I forgot yesterday, but I'm not going to forget today. On Blackboard, should still be there, I keep copying it over for years now, there is a video illustrating this very nicely. Okay, I don't know if you folks have ever seen, it used to be called like Kazur Kazat, now it's called In a Nutshell. They do these beautifully animated short videos. I posted a CRISPR one earlier in the semester. They did one for the immune response. And they cover both chapter 16 and 17. They talk about all the different cell types, and they try to show as many of their roles as they can. So going from cut through these various lines of defenses, done in like 10 minutes. Okay, I highly, highly recommend watching it to kind of get through these few chapters, because it really paints everything together beautifully. So they show the dendritic cells at work and how they bridge the two responses. Okay, and then the lymphocytes. Lymphocytes is everything chapter 17. These are your adaptive immune cells. B cells, T cells, and then technically natural killer cells, which I don't have listed there. They are not phagocytic. As we're going to find out, they have very highly specialized roles. And we're going to spend a lot of time talking about these B cells and T cells in the next chapter. All right, so those are agranulocytes. And I'll try to reference back to these as often as I can so you can kind of see the roles. All right, so getting to phagocytosis now, because now we know there's a number of those cells that are phagocytic. Let's see how phagocytosis aids in combating pathogens within the body. Okay, so this is a process in which your cells engulf and digest foreign bodies, killing them. Okay, so when you have an infection, the white blood cells go to work cleaning the wound. Now, as we saw, not all white blood cells can do this, and very few cells in your body can actually go through phagocytosis. So, the list of our primary phagocytes: neutrophils, eosinophils, macrophages, monocytes, and dendritic cells. Okay, basophils get kicked off the list. Mast cells, which we don't talk about, get kicked off the list. Lymphocytes don't have to on this list. All right, so these cells are really good at finding pathogens in your body. Okay, so we're going to lean on the example of macrophages. Okay, and I do that because macrophages are kind of omnipresent in your body. They're kind of found everywhere. They're only one of your white blood cells is found everywhere. So they're constantly scavenging your body for things that don't belong there. So macrophages, and most of these phagocytes, they're bristling with receptors. Various scavenger receptors that let them kind of sniff out various pathogenic chemicals. Right? They got to be able to find the pathogens in your body, which means they have to be able to sniff them out and find them. So imagine it's just bristling with all sorts of receptors. We'll talk about these more in a bit. Okay, so place a lot of emphasis on macrophages because, again, they're very important. You have two major types. You've got fixed and free macrophages. Let's do it out of order. Let's talk about the free macrophages. They're free because they're free to go anywhere they want throughout your body. Okay? These guys get into the deeper tissues of your body. They get into your central nervous system. They're the only ones that can do this. Okay? Other white blood cells cannot. Now, fixed macrophages, on the other hand, are fixed in three major areas in your body. They are found in your lungs, which is important given how dirty the air is and how much you're breathing every day. They're constantly killing all the stuff you breathe in. They're found in your liver. Right? You don't want infections in your liver. That would be quite bad. And you have a healthy amount of them in your central nervous system. And this is really the only white blood cell to be found in your central nervous system. Okay, so they're there to help protect you against things like meningitis. Now, well, I'll talk about that last bit here in a second, but they have the ability to call upon other white blood cells if necessary. Okay, so the steps of phagocytosis are not too terrible. So, first step, find the pathogen. So, when you have a pathogen in your body, be it a virus or bacteria, they shed. Okay, they shed things like peptidoglycan, LPS, various proteins. They leave chemical trails. Macrophages and other white blood cells can follow these chemical trails. So, I picked up some pathogenic peptidoglycan, I'm going that way. Okay, that's good old-fashioned chemotaxis, right? Moving towards or away from some chemical stimuli. We talked about that back with Flagella. So, like bloodhounds, they track down the pathogen. What next? Adherence. They need to grab the pathogen. So, there's a number of other receptors on the white blood cells that allow them to grab onto pathogens. So, for instance, you can see here, it's using whatever that green receptor is to grab onto the surface of that bacterial cell. Now, some pathogens can defend themselves from this stuff. Remember capsules from way back when? A type of glycocalyx, a sugary coat. Capsules resist adherence. It makes them slippery. But there's a way around that too. When we get to the complement system, we're going to talk about opsonization. There's your word of the day, opsonization. It's a fancy term for coating. We can cover the capsule, making it sticky again. So, there's always a bigger fish, right? All right, so find the pathogen, adhere to the pathogen. What next? Ingest the pathogen. So this is a type of endocytosis. And in almost all forms of endocytosis, if I'm not wrong, your cell wraps itself around the foreign body. So it's engulfing it. And when it finishes engulfing, it creates a vesicle, a membrane-bound organelle, called a phagosome. This phagosome here is effectively a prison cell. It's a holding cell for what's about to come that's way worse. So how does digestion happen? How do we actually break down the bacteria? Well, that involves another vesicle, lysosomes. Okay, so your white blood cells have a lot of lysosomes in them. So this diagram's not the best because it's got one sad lysosome. There'd be a multitude of them, many of them. So this is what transpires. The lysosome here, which is full of all sorts of nasty, degradative enzymes and chemicals, will merge with the phagosome. Okay? Membranes can do that. They can merge together. And when they do that, they dump those digestive compounds into the phagosome. Now, when enough of them do this, we create something new. A phagolysosome. Right? Play on words. You combine the two vesicles, you give it a new phagolysosome. And that's when digestion occurs. How does that actually happen? Well, let's illustrate this with 4 different enzymes. Lysosomes contain, 1, proteases, enzymes that break down other proteins. They will contain lysozyme, our favorite enzyme that breaks down peptidoglycan. They will contain nucleases, enzymes that ruthlessly break down DNA and RNA. They will contain lipases, enzymes that break down lipids. So, there is really nothing safe in that bacterial cell that's a macromolecule. It's all fair game to be broken down. Now, some bacteria can resist this, so we can step it up. We can also produce peroxidase, an enzyme that puts hydrogen peroxide into this vesicle. Okay, and as we all know, hydrogen peroxide is bad for cells. And you can even get fermentation within these vesicles, causing the pH to become acidic. So essentially, we're creating the most inhospitable environment possible for a pathogenic cell. So, for most pathogens, this spells death. Some, though, can even escape this, as we'll get into with tuberculosis in the last week. Alright, so, we're left with this bacterial soup, which leads us to discharge. So, We get to exocytosis, where the vesicle merges with the membrane, discharging the remains. So, find the pathogen, attach to the pathogen, engulf it, digest it, discharge the remains. That's phagocytosis. Okay, and this is what macrophages are really good at. My fun fact. Macrophages can actually merge with each other, making giant cells, just huge macrophages. I believe this typically happens during parasitic infections. Alright, so, I've been bad about this. I haven't asked if there are any questions yet. I'm sorry. But any questions about phagocytosis or any of the white blood cells? Okay. Yes.
Could you mention again which white blood cells have phagocytes?
We'll say, how about which ones don't? So, basophils. So, those are the ones involved in allergies and inflammation. And then the lymphocytes, B cells and T cells. Those are the only ones off the list. Alright, now, cell communication. So, how does your body actually begin communicating that something's wrong? Okay, so we're going to look at this as three very overall steps. First, you need to see the pathogen. Then you need to send out chemical signals alerting the immune system. And then you need to recruit white blood cells. So, cell surface receptors, cytokines, and adhesion molecules. Okay, so let's say we've got a macrophage. And our macrophage... Our macrophage is covered in receptors. Now these are transmembrane proteins. They span the length of the membrane. So they face outside and inside of the cell. So we got all these receptors. And then inside, since this is a human cell, you have a nucleus. Okay. So, all cells have receptors. Okay, not just white blood cells, all cells have receptors. Our cells, bacterial cells, archaean cells, whatever. Receptors are how how cells see their external environments. So they can detect different stimuli. And each receptor detects a different type of stimuli. So what is the term that we give the actual stimulus? The molecule that interacts with the receptor. That's called a ligand. Super, super broad term. Okay, and ligand is anything that can interact with a given receptor. Could be oxygen. Okay, could be salt. Could be glucose. Or in the case of white blood cells, it could be peptidoglycan, flagellin, pillin, RNA, DNA. Okay, so our white blood cell is just bristling with these receptors, so it can see all sorts of different pathogenic ligands. So let's say our white blood cell here detects something pathogenic. So this receptor reacts, and reacts by sending a signal to the nucleus, telling the nucleus to start producing cytokines. Okay, so cytokines are very important chemicals that your white blood cells can produce. These are chemicals that elicit various immunological responses. So your body makes several different cytokines. Each cytokine elicits a different response. It sends a different message. And some of them can actually send multiple messages. So for instance, a macrophage can produce chemokines. When a white blood cell senses that you cut yourself and there's bacteria in there, it's going to want help. It's going to want other white blood cells to come and help it clean out the wound. So chemokines attract additional white blood cells. Now, colony-stimulating factors. Okay, remember the whole hematopoiesis thing with all the steps from stem cell to blood cell? Colony-stimulating factors are the chemical signals that tell those stem cells what cells to become. Okay, so there's various types of colony-stimulating factors, right? So that white blood cell there, that macrophage says this wound's infected, we're going to need more neutrophils to help out. It would send these colony-stimulating factors to the stem cells, telling them to turn into neutrophils. All right, now others we'll see later on or in the next chapter. Interferon. The interferon is the antiviral cytokine. Okay, you make 3 of them. We'll talk about them more next time, but they can be produced to prepare your cells for viral invasion. Interleukins, these are involved in triggering B cells and T cells. One of them, I never remember, is involved, there it is, 6 and 1, you don't have to know that, are involved in triggering fever. And then the scariest of the lot, tumor necrosis factor. Tumor necrosis factor sends several signals, but it is extremely pro-inflammatory. Okay? It sends such a strong signal that you can die from producing too much tumor necrosis factor. Okay, so for instance, when people are dying from Ebola and you see bleeding from the orifices, they're experiencing what's called a cytokine storm. Their body's last-ditch effort is to say, oh damn, I'm just going to make as many of these as I can and hope the person survives. The tumor necrosis factor causes internal bleeding, and thus the bleeding from the orifices. So it's not just a virus killing them, it's your own body killing themselves because of how powerful these chemicals are. Okay, so normally these are highly regulated. This triggered macrophage would produce them in a regulated way, eliciting the various responses. That leads us to adhesion molecules, which we'll see more next time when we get to inflammation, but the macrophage is in the wound, says, okay, they cut themselves with bacteria here, it makes cytokines, it attracts more white blood cells. So that begs the next question. How do the white blood cells know where to stop? Right? How do they know where the injury is? Because of adhesion molecules. When you cut yourself, inflammation causes it to bleed like crazy for a few weeks, right? You're like, oh, come on, stop bleeding. Part of that is recruitment. Okay, the blood vessels dilate so that white blood cells can help reach that point. The blood vessels produce adhesion molecules, sticky proteins, like little hands that grab the white blood cells when they reach their destination. Right? They're like, okay, you are at the wound, stop here, get to work, clean out the wound. So it's all tied in together, right? Receptors detect invasion. Chemokines elicit immune responses, say attracting more white blood cells. And then the adhesion molecules around the wound stop the white blood cells at their destination. It's all tied in together. So that leads us into sensor systems. Okay, so actually detecting when invasion occurs. So elaborating on, for instance, receptors. But any questions about cell communication? Okay, so sensor systems. So as we go through this, I want you to think if there's any word that we have missed inside of your body. Okay, because we're going to talk about receptors that are on your cell surfaces, receptors within your cells, and receptors that aren't even attached to your cells, just floating around. Okay, toll-like receptors. The first one's discovered. So all of these receptors on our macrophage are also called toll-like. And toll-like receptors are how your white blood cells can see their external environment. It's how they can sense pathogenic chemicals in your body. Okay? So ligand. Ligand is a broad term for anything a receptor can interact. This is a specific set of receptors. So there's got to be a name for these ligands that they interact with, these pathogenic ones. We call them PAMPs. Pathogenic associated molecular pattern. A PAMP is anything potentially pathogenic. Okay, so, skip to the next one. So your white blood cells, they're bristling with all these different toll-like receptors. Each one interacts with a different PAMP. So, for instance, and I like this one, I stole it from OpenStax, it's very straightforward. Say this white blood cell here has 3 toll-like receptors. It's got one for lipopolysaccharide, or lipopeptide here. It's got one for flagellin, and it's got one for peptidoglycan. Right? 3 different potentially pathogenic PAMPs. But as you can see, each one interacts with a different shape. a different PAMP. Now, most toll-like receptors face the outside of the cell. A select few, though, actually face the inside of vesicles, so they can determine if they've entrapped anything pathogenic as well. But this is how they sense their external environment, and when they do engage a PAMP, when one of these toll-like receptors binds, say, peptidoglycan, then the rest of the cascade occurs. The white blood cell signals to the nucleus to produce cytokines, and the immune response begins. So, these toll-like receptors are associated with primarily those phagocytic white blood cells. All right, now, NOD-like receptors. NOD-like receptors are a lot like toll-like receptors, but one big difference. They're not bound to the membranes of your cells. They're not facing outwards. They're actually floating around inside of the cell. But they do the same thing. You had a whole bunch of different NOD-like receptors. Each one interacts with a different PAMP. So, toll-like receptors are for detecting extracellular pathogens, pathogens outside of your cells. NOD-like receptors are for detecting intracellular pathogens. Some bacteria invade your cells directly. We know that viruses sure do, right? So they determine if your cells have been compromised. Now, in addition, NOD-like receptors are more commonly found throughout your body because they are also used for determining if your cells have been damaged. So they engage DAMPs, damage-associated molecular patterns. Okay, so if your cell's been damaged, it's becoming weak or old, the NOD-like receptors will recognize that, and they'll say, okay, this cell is done, let's kill it. So they trigger apoptosis. There's a fun gen bio term for you. All right, now, this is going to be the last thing I go through because I'm going to go a few minutes long. The most complicated sensor system we've got is the complement system. So, NOD-like receptors, toll-like receptors, they are associated with your cells. The complement system is not. It is free-floating proteins found primarily in your blood and lymph. So, the complement proteins, which I'll call the C proteins, are typically in an inactive stage. They are just floating around your body, naive, waiting to be activated. Okay? As we'll see, there's going to be 3 different stimuli that activate. Okay? 3 different types of PAMPs that can trigger them. So why is it called the complement system? Well, when they do activate, they perform roles that complement and assist the rest of the immune response. So they help out white blood cells. Okay, so in your blood, you have what's called the C3 protein. And it is a very big protein. Now, C3 can be activated in 3 different ways. So the first way is the alternative pathway. It's ironic it's called alternative because it's actually the most common method of activating them. We call it alternative because we discovered it second. Whatever. So, in the alternative pathway, C3 activates merely by bumping into a pathogenic cell. Right? It's covered in PAMPs. So bumps into it, says, oh, pathogenic entity, I better act. That was nice and straightforward. Now, the lectin pathway is a little bit more specialized, and to save on time, I'm not going to get as much detail, so we'll cut it short this way. There's this sugar called mannose, or mannan, same thing. Mannose is not a sugar you would normally find in the human body. It is a sugar, though, you'd associate it with many bacteria, especially gram-negative, right? The whole LPS layer. So, in the case of some gram-negative infections, mannose gets into your bloodstream. The short version is the C proteins recognize this. And they say, okay, there's mannose here, I better activate because there's something pathogenic around. Now, the classical pathway, which is the first one discovered by humans, the classical pathway involves something we haven't talked about yet, antibodies. So, when you have a pathogenic bacteria in your body, and you produce antibodies to target it, the antibodies just bind to that pathogen. Okay, so the little red stick guys here, these are antibodies. In the classical pathway, the C proteins bind to the antibodies. And they say, okay, the antibodies are working, we better get to work too. The C proteins activate that way. So we have three pathways of activation. Alternative, where they bump into the pathogen. Lectin, where they recognize mannose in the body. And then classical, where they bind to antibodies that have bound to a bacterial cell. So the funny thing is, it doesn't matter which pathway you pick. You end up with the same series of events. So there is a cascade that occurs, and I'm not expecting you to know, but I just want to illustrate here briefly. So I mentioned that C3 is a huge protein because it's made up of a whole lot of little subunits. So when C3 activates, it actually chops itself up into smaller pieces. It undergoes self-enzymatic cleavage. So you can see this cascade is just breaking down into smaller subunits. The smaller subunits have roles. They are active. So C3a can trigger inflammation. Okay? C5 breaks down into a number of subunits, like C6 through C9. They're involved in what's called cytolysis. And then C3b goes off and is involved in what's called opsonization, we mentioned earlier. Okay, so it's made up of a bunch of smaller subunits that perform three major roles. Okay, so what can these C proteins do? Well, one, some of them can trigger inflammation. Okay, I never remember these, I'm not expecting you to, but C3a, C5a, these will interact with mast cells and basophils. And they say, hey, start making histamine, trigger inflammation. The other thing they can do, which is a bit more ruthless, is they can directly attack bacterial cells. Now, there's one big restriction. Cytolysis is only effective against gram-negatives. So gram-negative. So these smaller C proteins, like C9 and C6 or C8, they'll form what's called MAC complexes. They will embed themselves in the outer membrane of gram-negatives, an LPS layer. They embed themselves, forming pores, openings. Now, here you can see three of them. I need to find an old diagram I had. I had a SEM image that shows a gram-negative pathogen just covered in these complexes. It's just riddled with holes. And that makes them very porous, leaky, until they explode, thus cytolysis, membrane breaking. Now, the third role is opsonization. Okay, this is assisting in phagocytosis. So as I mentioned, opsonization is just a really fancy term for coating or covering something. So C proteins will bind to the surface of pathogens, making it easier for your white blood cells to grab them. Okay, so an opsonized pathogen is much easier to phagocytize than one that is not. So, we have three pathways of activation. Doesn't matter which pathway you pick, you end up with the same three roles. They can help trigger inflammation. They can bind to gram-negative outer membranes, forming these pores, these MAC complexes, or they can just coat a pathogen, opsonizing them, making them easier to phagocytize. But I would argue all three of these help out with the help out with the immune response. So the last receptor here is a newer one. Okay, and it's going to seem like it belongs back with NOD-like receptors, but they are independent. We've got RIG-like receptors. Okay, I only started hearing about these guys less than 10 years ago. These are receptors purely dedicated to recognizing one thing: viral RNA. And that's important for our cells because most human viruses are RNA viruses. Okay, so you got a whole sensor system dedicated to just detecting these RNA viruses. So what I like to also add here, and why I do show this slide, is what happens when Rig-like receptors are triggered within your cells. So this is my chance to introduce you to another important cytokine. And remember, cytokines are these chemical signals that elicit different immune responses and different immunological signals. So interferon. This is your antiviral cytokine. So if a cell is infected with a virus, it will produce interferon. The interferon is secreted outwards, and it does several different things. So you have three kinds of interferon: alpha, beta, gamma. Alpha and beta essentially do the same thing. Alpha and beta interferon are secreted to the neighboring cells, the cells around the infected one, to warn it of a virus. Say, hey, cells in the area, just so you know, there's a virus around, get yourself prepared. And it will trigger those cells to start producing antiviral compounds. Start preparing just to deal with that inevitable viral invasion. Slow it down. Interferon gamma, on the other hand, is a cytokine that goes to your phagocytic white blood cells. So macrophages and neutrophils would receive interferon gamma, and that also tells them to prep themselves for a virus, but in a different way. They start making antiviral compounds like nitric acid, so that when they do phagocytize that virus, when they engulf it, they are making a compound necessary for destroying viruses, things like nitric acid. So just prepping them. So interferon is produced when your cells are infected by a virus and secreted to warn neighboring cells and white blood cells of a virus in the vicinity. All right, so that's it for the sensor systems. Last two bits here are inflammation and fever, and I deal with these very lightly because this is one of those things I rely more for phys teachers to handle. So inflammation is a part of your innate immune system, right? We all experience inflammation. We all know the cardinal signs and symptoms. So redness, pain, heat, swelling, right? That is inflammation in 4 symptoms. And it is uncomfortable. Okay, it is painful, and when you cut yourself, you seem to bleed like crazy for a little while because of inflammation. But these all have important roles in the immune response. So, why do we need inflammation? Well, it does 2 major things. 1, it localizes the damage. Okay, so it keeps the pathogens from potentially spreading. In addition, that increased blood flow not only prevents the pathogen from spreading, but it also recruits more white blood cells. Right? So the white blood cells can come to the site of damage and get to work. So while it is uncomfortable, it is important short term in dealing with injured areas of your body. Okay, so what can trigger inflammation? Well, we've already mentioned these. Okay, so 2 out of 3 of these are old news. So the first thing that can trigger inflammation, toll-like receptors. Okay, if a white blood cell, like a macrophage or neutrophil, detects that there's a pathogen in the area, it will make pro-inflammatory chemicals like histamine or tumor necrosis factor. Okay, so white blood cells can trigger inflammation. Okay, basophils alone, basophils' primary job is to trigger inflammation. So if they pick up pathogenic PAMPs, then they will also start producing lots and lots of pro-inflammatory chemicals. The complement system, right? We just rehashed over this. One of the 3 ways your complement proteins can assist the immune response is by engaging inflammation. Complement proteins will bind to mast cells and basophils, telling them that they need to start making pro-inflammatory chemicals. And then tissue damage, and this is where I lean on the phys teachers, tissue damage leads to its own crazy enzymatic cascade of events. Okay? Damaged cells will end up producing a number of different chemicals, including prostaglandins. Okay? Prostaglandins are released by your damaged cells. They are also pro-inflammatory. All right, so let's see very briefly here what happens during inflammation. So first things first, we're going to have to injure ourselves. Okay, so in the image you can see there's some kind of blade cutting the skin. Now, folks, unless you cut yourself with a perfectly sterile utensil, it's going to be dirty. There's going to be bacteria on it, and they're going to get in that wound. Okay, so when you cut yourself, assume some bacteria can get into that wound. What happens then? Well, there are resident macrophages not far off. Okay, they are under your skin, waiting for this to happen. So if those macrophages detect that the wound is dirty, they react. Right? They use their toll-like receptors to detect the bacteria in the area, and then they start making cytokines. Okay, they're listing all sorts of responses, including triggering inflammation. All right, so step 3 is where things get complex. So pro-inflammatory chemicals like histamine or tumor necrosis factor will bind to your blood vessels. And they will cause those blood vessels to dilate, to effectively crack open. Now, this is what we call vasodilation. And when you cut yourself, right, it seemingly bleeds for longer than it should seem like it ought to. Right? Like, come on, stop bleeding already. And it's tired of dabbing off all this blood. That's because of vasodilation. The nearby blood vessels are cracked open, they are dilated, allowing additional blood flow. So that part, again, is to localize the infection. Right? Try to keep the bacteria from spreading by bringing more blood. And 2, that's how your white blood cells are going to know where to go. Okay? One of the cytokines was chemokines. That macrophage that detected the injury is going to make chemokines. It's going to call for additional white blood cells. And they're going to follow that chemical trail. They're going to follow those chemokines back to the site of damage. Now, at the end of cell communication, we talked about adhesion molecules. Okay, sticky proteins that allow your cells to grab onto each other. So right here, the blood vessels surrounding the site of damage, they're grabbing on to these white blood cells. Okay, those are those adhesion molecules. We call that margination. Okay, so they grab the white blood cells when they get to their destination, the area of damage. From there, the white blood cells will then squeeze between those dilated blood vessels. Okay, we call that diapedesis. So they get to the site of damage, they squeeze their way out, and then they get to work. They do what they do. They phagocytize the bacteria in the wound, cleaning it out. So it gets a whole series of events, right? You injure yourself, the pro-inflammatory chemicals dilate the blood vessels, increasing blood flow. That recruits additional white blood cells. They are stopped at the site of damage, they squeeze their way out, and then they clean out the wound. So, many of you have probably experienced a cut that ended up being a little pussy, get that kind of yellowish clear discharge. That is white blood cells that have become exhausted. Okay, they have exhausted themselves to the point where they're dead, and that's in part that discharge. So the wound gets clotted, the white blood cells clean it out, tissue repair and healing occurs. Okay, so the stages here in writing, right? So we've got vasodilation, right? Cut yourself, resident macrophages produce pro-inflammatory chemicals, the blood vessels dilate, increasing blood flow. Second part here, phagocytes migrate to the site of damage. So they follow those chemokines. They use chemotaxis to work their way towards the site of damage. When they get there, they are grabbed by the nearby blood vessels. Okay. Margination, adhesion molecules stop them at the site of damage. Diapedesis, those the white blood cells squeeze their way through between the dilated blood vessels, and then they get to work. Right? Wound gets clotted off, the white blood cells clean out all of the bacteria, and then the tissues get repaired and they heal. So, food for thought, short-term acute inflammation is good. Okay, it's uncomfortable for a little while, but is obviously quite essential for dealing with pathogens in your wound. Now, chronic inflammation, on the other hand, is not so great. Okay, chronic inflammation can lead to severe tissue damage and scarring if gone on too long. Okay, anybody with arthritis of any sort will tell you that. All right, but any questions about inflammation? Yeah, same.
Why do we rely so much on using ice? Like, what's the role of using ice in like an acute inflammation injury? It clearly serves a purpose. Well, why do we use that?
So the tissue damage doesn't become too severe because inflammation can get a little out of control. So like if you have like a sprained ankle, they tell you RICE, right? Rest, ice, compression, elevation. You know, I'm talking like you cut yourself, that comes and goes in like 30 minutes. But if you've got soft tissue damage and that's going to be swollen for days, you got a subsided inflammation. Otherwise, it can lead to some scarring if you don't, if you're not proactive about it enough. It can prolong the healing process because it's so swollen. It can't heal as readily. I'm fudging through what fizz I know. I'm sorry, but that's the best I know. All right, so fever. Okay, another part of our innate immune system that we all experience the same. Right, it's that increase in your core body temp. You know, most of us get very uncomfortable. We don't like fever. We take Tylenol or ibuprofen to bring it down, but it is an important part of your innate immune response. So let's talk about what triggers fever first before we talk about what it does for you. So if you had anatomy or phys, you probably know that your body's thermostat is your hypothalamus. Okay? In your brain, your hypothalamus dictates that your body should be at 37 degrees Celsius, which is pretty toasty. Something has to tell your hypothalamus that it needs to start increasing that temperature. Okay? Usually just a couple of degrees. So that's what we call the pyrogenic response. Right? So heat-generating response. Now, there's two ways of doing this. Okay, so you get pyrogens, okay, chemicals that can cause this to happen. So the obvious ones are endogenous pyrogens. Okay, unsurprisingly, it's the cytokines. So the primary cytokine involved in the pyrogenic response here is interleukin-1. Okay, this is an endogenous pyrogen. As an infection continues, more and more interleukin-1 is produced. And as interleukin-1 accumulates, it gets to the hypothalamus. When the hypothalamus hits a critical point, it says, okay, we've got enough interleukin-1, let's start raising the core body temp a couple of degrees. Okay, this is where you get up to about 39 degrees Celsius, you know, your typical fever around a 101. You want to talk about Fahrenheit. Now, exogenous pyrogens are a little bit more troublesome. Okay, this starts as chemicals that are not made by you. Okay, these are foreign pathogenic substances. Now, how exogenous pyrogens work has been under debate recently. Okay, so for a long, long time, we thought these were foreign substances that went straight to the hypothalamus. Okay, that may or may not be the case, but it's looking like it's more of an issue where you have pathogenic substances that are causing your white blood cells to freak out. So, for instance, toxins. Okay, pick your favorite, whatever. Tetanospasmin, the botulinum toxin, pertussis toxin, cholera toxin. It doesn't matter. Many of these toxins get into your bloodstream. They're water soluble. When that happens, all of those white blood cells in your blood freak out. They're like, oh crap, this is something really, really bad. We're going to overreact. And they start making too much interleukin-1. And that's when you start getting those critical fevers. Those 105, 106, 107, and up. Okay, so often, if you get particularly high fever, it's because of exogenous pyrogens. Okay, your immune system is just freaking out over how bad something in your body is. Okay, if it's a more typical fever, it's probably endogenous pyrogens. It's just your own interleukin-1 doing what it normally does. All right, so we get this increase in our core body temp. It makes us miserable. We try to kill it off, but like I said, it is important. Okay, And folks, this is not a new concept, right? This hails back to talking about enzyme function. Okay, we had 3 graphs when we talked about enzyme function. Temperature, pH, and substrate concentration influence how enzymes work. So looking at that curve regarding temperature, right? As temperature increases, all of a sudden you see protein and enzyme activity drop like a rock. They denature. Right? Proteins are sensitive. If you heat them up, they fall apart. So, by increasing your core body temp, you are A, slowing down the pathogen's growth, so your immune system can catch up. And that slowing is also in part because you're starting to denature the pathogenic enzymes and proteins. Starting to break them down. Okay? So you take the pathogen out of its comfort zone. In addition, your immune system functions better at elevated temperatures. White blood cells function more optimally. The effects of some of your cytokines are more potent, like interferon. An antiviral cytokine works better at increased temperatures. And your body will also start making things like transferrins or ferritins. Now, we mentioned these last time briefly. These are proteins that hide free iron from pathogens. Okay? You wouldn't normally make a transferrin if you're healthy. You wouldn't need to. You'd make it in response to a pathogen that would be stealing iron from you, okay? Which you'd probably be experiencing fever during that time as well. So you take the pathogen out of its comfort zone, okay, start denaturing its proteins, and it actually enhances your immune response as well.